Welcome. Happy Friday. Slightly out of sync Apex session today, a special talk. Very pleased to welcome as our guest Dr. Jan Spacek from ALFA Mars, as well as a senior research scientist at the Foundation for Applied Molecular Evolution in Alachua, Florida.
Alachua, Florida.
Right, so Jan has a couple of really catchy, eye-catching titles, so I don't think you need to hear from me. Without any further ado, hand over to Jan, 45 minutes or so, some questions at the end. If you could hold your questions for the end, that'd be great. Folks online, just let me know if you can hear me, and same applies. Thank you, Jan.
Well, thank you everybody for coming. Although the semester hasn't started yet, as I heard. Apologies. So, at least I know that you are here, you are really interested in what I'm about to tell you. So, this title, I gave this talk yesterday at a Breakthrough Foundation or Breakthrough Discuss. So, this differs slightly from what I posted that I'm going to present here, but the topic is the same. So, it's Mars and Venus, clouds of Venus specifically. So, for now, almost a century, scientists were puzzled why Venus clouds are yellow. As you can see here in that picture, that's how you see it through a telescope. And in a UV, you can see actually very dark patterns emerging from that deep cloud. So we still, to this day, are unsure why. Why is this coloring? Because the cloud's supposed to be concentrated sulfuric acid, and sulfuric acid is even less light absorbing than water. So many scientists were trying to crack this puzzle, and still we call the absorber, the light absorber, the unknown Venus absorber. So how I started working on this, that was through Breakthrough, that's a Breakthrough Initiatives organization. I was looking for funding for my project to look for life on Mars. I applied for several grants on NASA and was rejected. And I talked to people from private research organizations or private funding agencies. And Pete Klupar, who was higher up in the Breakthrough Initiatives, diverted me instead to Venus because Breakthrough was at the time interested more in Venus. And so that's how I started in 2021. I got funding for me and one student for 2 months. And we wanted to find if some organics are actually fluorescent in sulfuric acid. And surprisingly, we found that a lot of organics, or maybe almost all we tried, were turning to fluorescent material upon putting it in concentrated sulfuric acid, which was very surprising finding. And while Gage Owens, my students, was working in the lab, I was trying to figure out what's going on regarding the chemistry. Then I proposed this organic carbon cycle in the atmosphere of Venus. And then the data we gathered were used to tune the laser in that little probe. So Morningstar mission to Venus is going to be the first private interplanetary mission that will fly hopefully next year. And it will carry autofluorescent nephelometer, which is a little instrument that shoots laser into the droplets and observe reflected light. If there's any fluorescence in there, you will observe the light with a longer wavelength than we put in. And so, yeah, this is, we refined with colleagues the original proposal for organic carbon cycle and showed how you can get from a simple photochemically generated organic material, or actually carbon monoxide and formaldehyde, to large organic molecules, which was very surprising. Until that point, the assumption was that there is basically no organics in the clouds of Venus because the sulfuric acid would destroy it, which we find it's not true. And then I continue thinking about the issue. before I get to it, I need to introduce you to this nice picture. You can see that the river is quite dark, but as it falls into the waterfall, as it breaks to small droplets, it appears brighter and brighter to the point where you have the mist, which is composed from the same dark liquid, but it appears completely white. The same effect you can see on this beer, right? The liquid is yellow, but the foam is white. And it quite doesn't matter much how dark the beer is, the foam appears much whiter. And so, speaking of beer, some of you might know beer-Lambert law, which is observation that molecules dissolved in water can absorb light, and from the amount of absorbed light, we can estimate or calculate the concentrations of the molecules or solutes in the solution, which is super interesting. But we cannot do it from the foam, right? Or from the droplets. We need to do it from the bulk liquid. And so, more interesting question than asking why is Venus pale yellow, as Carl Sagan was talking about it. It's a quite important question to ask: how dark actually is the liquid which forms the clouds? And from like these everyday observations, you know that the smaller the particles, the more scattering you have. So they appear lighter and lighter. And we know that in Venus clouds, you have sub-micron particles as the main peak in the distribution. So most of them are very small. So the scattering effect is huge. And so this suggests that the liquid must be actually very dark, because we see it's slightly yellow. So it has to be super dark to sort of at least like, but this observation is quite poor, right? From this, just using this logic and observation, we cannot guess if it's dark as the Guinness I showed you, or if it's as black as a tar. But I was lucky to talk to this lady who works in South Korea, and she's a specialist on a radiative transfer model and modeling the Venus clouds. And she helped me to model actually how efficient the absorber in the liquid which forms the clouds must be to make the Venus appear yellow as we see it. And so this is the data we will publish next week, hopefully. This was in the making for quite a long time. But what's very interesting about this is the units on the axis is absorbance per centimeter, and notice it goes almost to 3,000. And August Beer tells us that the absorbance is logarithmic. It's a ratio between the incident light and the transmitted light. And so if you have absorbance 4, means that you have 0.01% light is transmitted through 1 centimeter, right? And if you have absorbance 3,000, then you have like almost 3,000 zeros, and then you have 1%, like 0.0001. And so this is extremely efficient absorber. And second thing, from the absorption per centimeter, if we know the extinction coefficient or absorption coefficient, that's the epsilon, we can know the concentration of the absorbing material. And second thing, we can compare this to some liquids we know. I actually suffered from for the science here. I pricked my finger, diluted my blood 1,000 times, and measured absorption spectrum of my peripheral blood, and then multiplied back to 1,000 to figure out what's the, compare the absorption of my blood, a liquid I know quite a lot, to Venus clouds liquid, and you can notice that the Venus cloud is actually 3 times more absorbing at the maximum over there than the blood, which means that you need to have a lot of absorbing material. As I said, this is very efficient pigment. Here's a comparison to some absorbers. might know, and the extinction coefficient. So these are like the best absorbers we have on Earth. Chlorophyll and heme B, those are like porphyrins. You notice that they need to have those double bonds next to each other. It's called conjugated double bonds, which are responsible for the light absorption. So they have the absorption coefficient, that's the epsilon, molar per mole per centimeter. It's like in thousands to 100,000 for those porphyrins. And so you would need surprising amount, like 10 to 30 grams per liter of these highly absorbing molecules to make the same absorption we modeled in Venus, which is extremely surprising. Now it's not the question, why is Venus pale yellow? The question is, why is there so much absorbing material? So another interesting thing, what our model for the first time quantitatively showed, we basically exclude the previous favorite absorber, which was suggested, which was ferric chloride, that little molecule over there. It's simply not absorbing enough, even if it's 100% concentration. You will need more than 100% to absorb enough with that at that maximum model. Another thing, astronomers don't like to go to lab. If they actually put the ferric chloride in concentrated sulfuric acid, they will notice that it immediately reacts and produces the sulfate. So the literature is doubly wrong. And not only it's not absorbent enough, which they didn't know before, it also reacts forming that white precipitate. So especially if you are aspiring astrobiologist or whatever science you do, don't stick to the boundaries of your science, just test things for yourself. And so here's a comparison to some chlorophyll, which fits quite a spectrum. So we can find examples of chlorophyll which match the spectrum we observe in the range we modeled. The thing is that in sulfuric acid, this might not be stable. We are not saying that there is bacterial chlorophyll in Venus clouds. We are just providing this as inspiration for how it would look like, and that you need about 30 grams per liter of this highly efficient material to absorb that light. Another interesting thing, if you put any organic material in sulfuric acid, it turns fluorescent, but it over time decomposes to this brown or black sludge. It turns to tar because there are a lot of reactions possible. So if you have complex organics in concentrated sulfuric acid, you would expect it to be black, and the spectrum would go through entire visible range. So visible range goes to 800 nanometers. So you will have like smooth frame because we have that sharp, because of the shape, we can know that this is the absorber is actually a specific molecule load. And so now collaborators who helped me with that, what I just presented, it's Jan Petkovsky and Jan Joli. They really liked the model from the start, the beer model, and I need to convince Paul Rimmer to convert him to our beer model. So last mystery about the Venus. What I presented is it's not too difficult. Quite simple concept. Basically, high school students can understand that if you have that waterfall, that the scattering effect increases. So I think that the reason why Americans or other astronomers haven't came up with this model before is because the American beer looks like that. There's no foam, so they have no chance to make the observation. I spent a lot of time making. And so, as I showed you, the Venus is very interesting, but it is not urgent. Yeah. Now, there was some controversy about Venus that we had some phosphine in the Venus clouds and parts per billion above the clouds, parts per million in the cloud estimated. And compared to what I just showed, that would be easy to explain. So you tell me how you can make 30 grams of highly absorbing, 30 grams per meter of highly absorbing material, very likely defined organic molecules in the clouds. But it's not urgent. What's urgent is something else. So the astrobiology right now looks like this. You know, you have the exoplanets, you have Venus, ocean world, and astrobiologists are, you know, like it's in the candy stores. That's what we are playing with because it's new and cool. But yeah, our results, we just put the bow on the Venus, so it's even more interesting. But there's a Martian biosphere, which is more urgent, right? Because unlike the other things, this is something we need to deal with right now. So now switching to Mars. So the more we know about Mars, the more we expect there to be a active biosphere. It's both from what we know about extremophile life on Earth, that different life form can survive in conditions which are approximate, like model sim conditions on Mars, and also how Mars looked in the past and how the conditions look now. So in 2019, there was a conference in Carlsbad, and a lot of people just agreed that, or a lot of attendees of the conference were convinced that extant life on Mars, it's likely. And so for decades now, a lot of astrobiologists were urging funding agencies to do something about it. We need to look for life on Mars, right? But it's quite bleak. There are a lot of other things we can do, and sending missions to Mars, they were quite expensive. So you have some sort of democracy in a decision making. You can feed a lot more scientists if they do observations of exoplanets compared to when you send a mission to Mars. So it was actually this political struggle, which led to the fact that we are still not looking for life on Mars. So when NASA tells you this, don't believe them. We've not been looking for life on Mars in a way that would be efficient. So the instruments right now on Mars would not be able to find life at concentrations we expected to be there. This was published a couple of years ago. Analysis, they took the instruments which are on Mars right now and which are planned to be sent, or which were planned to be sent 2 years ago, including ExoMars mission, and none of the instruments was sensitive enough to pick up signals from sparse life in Atacama Desert, because when you have just 100 cells per milliliter, you need very sensitive instruments, and they just couldn't see it. So if life on Mars is a little bit different from what we know now, the instruments would definitely miss it. So, and why it is urgent to do something about this right now? Because we plan to go to Mars. It's not just current administration, it was long in planning. The previous administration also promised to send humans to Mars before 2040. And also Chinese Space Agency and SpaceX. And I'm sure that other nations and maybe private organizations will go to Mars once it's economically feasible. With advance in rocket science, it will be feasible quite soon. I believe we have the last decades before decades is optimistic decade. It's quite probable that within 10 years we might have humans on Mars. And so, yeah, this is Chinese Chinese plant, but Chinese space agency is quite mysterious. They don't release much information, but this is the latest prediction what they are going to do from the official release. Notice that Venus sampler return in 2033, so they maybe bring the dark material I described, but the Mars science station in 2038. Before they make the station, they need to do some preparation as well. So we'll see. So in astrobiology, it's not just science; it's also combined with politics. So you need to count on that. So speaking of politics, the guy in the middle, that's Robert Zubrin, the president of Mars Society. Guy in the blue shirt, the dark blue shirt is my boss, and we wrote an article in 2022, how to search for life on Mars, and that's the important line. First, stop refusing to look, because that's what we in the whole world, that's what we've been doing so far. In that article, Robert argues that there's not much danger from Martian biosphere to Earth, because argument is that when you have specialists on Mars, it's just like sharks, whereas here we have like lions in savannah. So there would be no competition because we have different environments. But of course, we know a lot of counter arguments. If you have a more advanced generalists, they can outcompete local specialists, like the case of the rabbits in Australia or rats in New Zealand, or actually rats all over the world. So Stephen is very pragmatic. Which position is more likely to get us more funding? So we need some risk mitigation insurance policy. There is currently no investment into a search for life. Actually, the only grant on the subject was received by us, and that was pennies compared to what needed. So that's how we do make the estimate how much we want to invest into it. So you will make your favorite disaster cost. Let's say that Martian biosphere will spread in our permafrost, release all methane within the next couple of centuries. How would that affect us economically, and multiply it by probability, and that's how much you should invest into searching for life on Mars. And of course, the other probabilities. NASA actually made this calculation. I hope they did, because they, for planetary protection reasons, they made a break the chain of contact for sample return. That means that nothing which touched Mars would touch Earth. So you have those samples in the tubes, which were collected, put in a box, and then in the spacecraft, they would be put in the other box without any contact to Earth, and then they would be brought to biosafety for sample return facility. And together, these two procedures, which are for planetary protection, would increase the cost by about 10 billion on that order of cost. So that's how much NASA is willing to pay for planetary protection. I guess based on that calculation, but know that sample return was cancelled, and there is no planned planetary protection for crewed exploration. We don't know what's you cannot do planetary protection if you do crewed exploration, because you cannot sterilize humans in autoclave. So there's currently no insurance policy. And so that's why the search for life currently living on Mars is the only space exploration endeavor that cannot wait. We need to act right now. So the solution is to go there and find life in situ before we send humans there. I don't believe that the alternative that we will all agree that we are not sending humans to Mars would work. Also, I want to go to Mars. I think it's going to be a great step forward for humanity. And so, yeah, let's go there, but be smart about it. So we need to succeed with this. We need to have an ability to find an unknown life forms, because Martian can be different. They can have different biochemistry. We have no idea about their ecology, which means we don't know where to look for them. And just because this is an assumption, because you have low energy environment, you expect low concentration of these potential Martian cells or Martians in general. And we have very short time to do that. Here I said 3 to 5 years. It's not because I expect humans to be on Mars within 3 to 5 years, but I would like to have some wiggle room to, if we find Martian biosphere, to study it a little bit more. And yeah, this sounds hard, but I think we have a solution which is much less costly than NASA wanted to invest into, 1,000 times less than NASA wanted to do the planetary protection for sample return. So $10 million. And so the solution was in a plain sight all along. This is Agnostic Life Finding Association Mars, where we propose to do just that, determine whether Mars hosts active biosphere before we send humans there. And to understand what we want to do, I need to explain something about the Martian water cycle. Mars doesn't have a large moon, so it's a rotational axis. It wobbles quite a lot. So when the obliquity is high, meaning that the sun is shining straight to the poles, the ice from the poles actually it snows on the other side, quite close to the equator, and the glacier basically spreads towards the equator. And then, because you have much higher humidity on Mars and much higher pressure, because all the carbon dioxide is sublimated from the poles, put on the other side, but you have much higher pressure. Estimates are about 100 millibars, which means that that's enough to keep water, liquid water on the surface. Which means that in spring, during high obliquity periods, you have wet surface when the snow melts. And so during those high obliquity periods, very likely the conditions are much more fertile, much more clement for life, any form likely. And so here's my lasagna ice hypothesis. In that mid-latitude ice, which is deposited from the about 40 degrees north and 50 degrees south towards the poles, which was the ice was deposited in layers of snow and dust. You would have samples basically from the entire planet. So just like we can find microbes in glaciers which were blown over like on Earth, we would very likely be able to do that on Mars. And we are sampling entire Mars from the time when it was much more clement. So this is the lasagna ice hypothesis. During the high obliquity period, the life was deposited in those glaciers. And then it's there and we can find it. The second ingredients into our project is that before we send humans to Mars, we will need to do some in situ resource utilization, which is mining local resources to make propellant for return ship. And also you want some water for your astronauts to drink and use for irrigation, whichever. So there will be very likely mining before we send humans there. And the mining, the most advanced approach towards the mining is done by Honeybee Robotics. Actually, now it's Blue Origin because they were bought. But you have this, there's a rig, you make a hole, you pump hot water down the hole, and then you pump it all up, and then you repeat the cycle. So you are making this well, they call it rod well. And so you will need to extract hundreds of tons of Martian ice to refuel those big starships. And so that's the next ingredients into our project. We plan to put our instruments onto these rigs. And another thing you need to know, it's a polyelectrolyte theory of the gene. Stephen Banner, again in his traditional blue shirt, next to his, Stephen works on synthetic biology. So this is example of DNA with more letters than 4. That's what we are working on in Florida at Foundation for Applied Molecular Evolution, synthetic biology. That's a different topic for now, but important part is that Stephen made a theory that all life, as we know it or don't know it, as long as it has to propagate between generation, you will need to have a molecule which stores genetic information. And to store the information, you need the molecule needs to be long linear polymer, and to enable the polymer to be, if it was without charge, it would collapse into a globular shape when you have charge on the backbone, like so. It stays spread out in solution, and it's much easier to spread and replicate it. So when you have genetic polymers, long molecules, they cannot be produced on current Mars abiotically. There's simply, the chemical conditions are not conducive to it right now. And life cannot exist without genetic polymers. That's the polyelectrolyte theory of the gene. And so this avoids false positive signals. For example, when you see methane on Mars, it can be produced through serpentization, for example. So we would not know if it's produced by life or not. Now we had some observation of dimethyl sulfide in exoplanets, and again, we are not sure if it's made by local life or if it's made through some abiotic chemistry we are don't know yet. So those would be potentially false positive signals, and you have, of course, potentially false negative signals. If you have assay to look for example, cellulose or some specific lipid, and you don't find it, it just might mean that Martians are not using it. So you might miss Martians if you are not looking for molecules that are universal for all kinds of life. So genetic polymers, which are polyelectrolytes, and which must be, store the genetic information in a sequence of building blocks. This is a Schrodinger's apereo crystal. And so if we find them, we found life, and we can be extremely certain about it. And finally, our instrument, now putting the ingredients together. So you have the mining on Mars. You have, we are basically taking the polyelectrolytes from a solution, and we concentrate them, purify them, and extract the polyelectrolytes. So, and then we analyze them on site for the biopolymers. We know we would use biological nanopore, perhaps PCR combined with the biological nanopore, which is a device to actually sequence the DNA, which tells us the order of those building blocks. For agnostic, again, agnostic not in religious way, but agnostic in a way that we don't presume the chemical structure of the molecule. We would use a solid-state nanopore, which is just basically a tiny hole in a membrane through which you are threading the polymer, and you are just studying basically the shape and the rough structure of the molecule, which again, if you find a long charge polymer on Mars. It's a smoking gun that we find, like, of course, it would be best. It would be best if we could also send the samples back. The thing is that we are also refueling the rocket, so it can bring the samples for more analysis. And we are not proposing to send a separate mission, which would be super expensive. We are proposing to add this instrument to missions that must fly to Mars anyway before we send humans there. So it's like no-brainer. That's something we should do. So this is how the instrument works. You can imagine it as a stack of membranes between two electrodes. And you are pumping the Martian water here on the top. And we first run it through a sonicator, which disrupts the cells. In case Martians have some cells or some compartments to release the polyelectrolytes into solution from which we can then extract them in the electric field. And so polyanions like our DNA would go towards anode, polycations like alien, polyelectrolyte or some proteins would go towards cathode, of course. Small ions would continue over the nanofiltration membrane, the membrane, the MFM, it's a microfiltration membrane. And yeah, we will not go into more details. If you are interested, we can discuss more details of this instrument later on. And you might see this guy over there. So Chris was helping me in summer 2022 with the first prototype of this. Then I had Kate visiting us from Princeton, and last year I had a bunch of students. Chris returned back, although he didn't spend much time in lab. He was more focusing on the PR and writing some interesting articles on primordial school. And so last year we got NIAC, NASA Innovative Advanced Concept Grants to develop the instrument which I just showed you. And this is how it looked. Doesn't look very glamorous right now, but it works. So it can separate, isolate and concentrate DNA from solution in a continuous fashion. So that proved that it's possible. So this is like desktop breadboard prototype. And we have a company in Czech Republic working on a more better looking and more professionally looking instrument. They actually already made a main part, the stack of membranes. And now they are working on that unit where you have, all the junk I have on the table. It will be nice, neat in one place. So just bigger and nicer. And okay, important part, we are targeting 2028 for the development. So it will be finished and space ready by 2028. I think we can do it. We get funding. And yesterday, I talked to Pete Warden. He asked me how much I need. And maybe we'll get some private funding, if not from NASA. So maybe we might be hopeful about that. So that's a presentation. Yeah, I now hope that you understand why Mars is urgent. We need all hands on deck. It would be nice to have more than one instrument to look for life on Mars, some orthogonal way how to show that, well, to find if Mars has a active biosphere or not before we send humans there. We have very short time to do it. There's a couple hundred people in the world who can work on that. So if you can work on that, you have some personal responsibility to do that. The funders should support the scientists, and let's not be stupid. Let's determine we have active life on Mars before we get there. So Chris, you might know that guy next to him. He's famous in the United States, maybe not in here as much. It's Bill Nye, Gabi Rizzo. She's doing fine now. She had some medical issues, but she's fine. And she's helping us a lot with ALFA Mars. And I have a lot of other people helping us, and you're welcome to help us as well. And Yeah, so join us, ALFA Mars, and other slide. Yeah, something to think about. This is what I put on after the yesterday conference, the breakthrough discussed was focused on life and consciousness and searching for life and consciousness in the universe. But the truth is, we are not interested just in life and consciousness. But you have examples where, imagine you have a swarm of von Neumann probes. You know what von Neumann probes are? That's the fourth experiment that you have self-replicating robots roaming through the galaxy, just sensing everything. So they might not be conscious, they might not be live, but we would be still very much interested in know something about them. And we are not interested, like when you are looking for life, you might not look for life, but you can still find it. So, for example, the example with the Venus clouds, like I have no idea how it's produced, and I was not searching for life. There you go. And another last examples. For example, there is still raging debate whether viruses are life or not, and whether virus is life or not is the least important thing you can know about virus, right? There's a much more interesting thing you can find, like how it spreads, how to defeat it. So again, life is not important for virology, and I think that life is not important for astrobiology. We just need to look for anomalies. And with that, it's 14 minutes. Beautiful. Please ask questions. Thank you.
Wonderful, thank you, Jan, for that fascinating and motivating presentation. Any questions from in the room?
Disagree. I hope you don't mind if I ask this or just bring it. He said life or consciousness. So I think the walls are fed up to hear this, but once we were looking in a pool and there was a stone. So, and the question was, why are you looking at the stone? Is the stone communicating with you? Is something from up there communicating? It wasn't, the stone's not alive, but is it conscious? That was a question of the older person. And because you've said this, we are not interested just in life or consciousness. And what Von Neumann said, could it be that dust is conscious from Venus or Mars, and that has life in it itself in another way beyond what we comprehend?
Yeah.
Because I said, what are we talking about? I'm
Okay.
looking at the stone.
So the question was, is dust conscious?
Yes.
And my answer is to it, I'm not interested.
Yeah.
Right, that's not a question for me, that's a question to debate with philosophers. I'm looking for practical things, and debate whether dust is conscious doesn't seem practical to me, so I'm very pragmatic in this sense.
No, I agree, but it's a
Yeah, that's a good question, but for somebody else.
It was a fascinating talk. Can I just find out your reason for the haste? Is it because you are worried that we will bring Martian biosphere back to Earth, or that we would destroy the Martian biosphere before we got a chance to study it?
So I'm going to repeat the question so everybody
Yes.
can hear it.
Yes.
So the reason, what's the reason why we need to know whether there's a Martian biosphere before we send humans there? Is it because of the forward contamination, which is contamination of Mars with Earth biosphere, or backward contamination of Earth with Martian biosphere when the astronauts come back? And I'd say it's both things. And what I'm most worried is things we don't know about yet. So going blindly is the worst thing we can do. So both of those arguments, we might find very interesting things about Mars, and I think we should make informed decisions instead of going blindly. Please go ahead.
Your idea of piggybacking on a mining operation, how sure can we be that all of the mining material and for that matter, your ALF, have been so thoroughly cleaned on Earth that there is absolutely no DNA on it at
Yeah.
all.
So again, repeating the question.
Yes, please.
How can we be sure that our instrument we are sending to Mars is going to be completely sterile? The answer is we will not be sure. We will probably find a lot of contamination we will bring with us. The contamination will be on the same level whether the instrument is there or not, right? As long as you are mining, you are bringing much more of the bacteria on the mining rig. And so the instrument will allow us to monitor the contamination we are bringing with us. And because we are actually sequencing the genomes, especially if you are sequencing genomes we already know, we will use metagenomics to analyze bacteria. And so if we can put it on Earth, the phylogenetic tree, and see that they are connected quite recently, there will be, or if it's the same as in Florida, you know, we will know that those were the stowaways we brought with us. Yeah, good questions. Thank you.
Any questions online, if anyone has any? I had one, well, I've got a couple actually, if there's no one else in the room. Again, how far, how deep, how long do we look before we accept the null hypothesis, right?
Yeah, I was just.
Sorry, it should come back in a second one.
Yeah, how long should we look before we give up on searching?
Or
Like,
accept the null hypothesis.
right.
I don't want to say give up, but you know, at some
Yeah,
point
yeah.
we're going to have to draw a line and say we need to come to some conclusion, especially if there's some haste involved.
Yeah. I don't know.
One-
So the question was, to repeat again, how to determine basically the upper limit we are looking for, right? We will say the answer which our instrument will give us, we will say there is less than 1 gram of DNA or polyelectrolyte per 1 ton. And if that's enough, right? And for that, I don't know, I think we'll try to get as large sample as we can. We are not very limited by the size of the samples because they will be mining thousands of tons. So the bigger the rocket, the better chance we have to find actually indigenous life. So that's advantage of sending those huge rockets, surprisingly. And if you have small concentrations of life and you are looking for just 1 milliliter, it's quite possible that you are looking on a wrong milliliter because, you know, the cell can be right here. And just because the life is sparse right now, it doesn't mean that, you know, when Mars tilts again, it might start proliferating quite rapidly again. So as long as there are a few cells, it can spring back.
And maybe following, sorry, Tim, just following on from that, if the samples of water, given the Martian hydrological cycle, they won't be discrete, right? So we might not be able to necessarily connect any indications of life with the local biosphere or a source necessarily. We'd be sampling a hemisphere or Mars bulk atmosphere, right?
Yep.
So where do we go from there? What's the next step? We find something interesting. Do we go local? Or do we stay global?
Yeah, both. I don't know. I'll tell everybody, and then it's up to you to decide what's the next step, right? Yeah, I would like to include everybody into this. So I think if you say news with a confidence that we found life, whole world will just go for that anyway. So then everybody can, you know, do whichever experiment they want.
How would you know if the polyelectrolytes or the DNA collected is from extinct or extant life?
Another great question. So how we can distinguish between currently living versus extinct life, which is already dead for quite a while? To that, the answer is it's like a feature. The DNA and polyelectrolytes, they are not very chemically stable. So if you kill the life, DNA in deep ice on Earth has a half-life about 1 million years. So within 10 to 100 million years, if you don't have active life, you will lose all the traces of those polyelectrolytes. So if you find life, it's polyelectrolytes. It's either from life which was living just before we suck it up into our instrument, or it was living within the next few million years ago. So, and then you can make assumption that our climate didn't change much in a couple million years. So you can make assumption that life is still active there. Great questions.
I did have one more. Maybe it was a trick one. So you mentioned that perhaps our current life or astrobiology missions that are currently on Mars might not be have the best instrumentation to make these determinations. But I think it was a paper, what was it, Sharma a couple of years ago, that suggested actually the Sherlock instrument picked up quite a diverse array of organics. So maybe suggesting preservation was better than we thought. Is that consistent with?
Yeah. So the question about sensitivity on the instruments and a question about sensitivity of Sherlock instrument, which found actually quite a lot of organics. Lately, we found those long hydrocarbon chains, which is super interesting again. Again, this is not an ambiguous sign. Again, we found probable signs of life. The issue with that is that, yes, the instruments currently on Mars can find organics, but they need some threshold concentrations, which corresponds to about, let's say, 1,000 cells per milliliter. If you have like 100 cells per milliliter, you would miss it. And that was the test I showed you, or the paper I showed you, where they actually took samples from Atacama Desert, where they used metagenomics to actually analyze the local microbiome in that ancient dry soil, which is analogous to the soil on Mars. Although it's even more fertile, let's say, but still you have about like 100 cells or less than that per milliliter, and those instruments would not pick up those signatures.
Well, unless any other questions, I think we could thank Jan again. That was fascinating. Great questions. The recording will be available, I think, in a week's time. And we don't have an Apex seminar running this semester, so we'll start again next academic term. But if you have any colleagues or collaborators that you think might be interested in presenting next term, please do put them in touch with me, and we can get them on the schedule. And hopefully see you then. Thank you so much for coming along. Thank you. Thank you.